Hi everyone, welcome to Basic Science Series. I am your host Kumar, and today we will discuss polymerase chain reaction part 3. In part 1 and 2 we have discussed the basics of PCR. We have discussed what is PCR, why it is important, what is the principle of PCR, what are the different components of the PCR, what is the general layout of the PCR. We have discussed how many things are required for polymerase chain reaction. In this particular video, I'll focus more on the primers. I'll focus how primers they're going to bind to the DNA strands, what are the different properties of the primer, when we use primers, how we're going to design them, what is the sequence of the primer, if you have a example sequence, how are you going to design primer to that particular sequence. So all those important things that we are going to discuss, and what should be the length of the primer, and what should be the basically length of your gene if you want to amplify, or the region that you are trying to amplify. So these things we're going to discuss in this particular video, and I hope that this video is going to give you additional knowledge on PCR and specifically on the primers. So without any delay, let's start the video. All right, as always, I'll start with writing the topic, which is polymerase chain reaction, and this is the part 3. In this particular part, as I've already told you, we'll be discussing the primers. So this is the part 3, and here we're going to discuss designing the primers. Primers, they are also known as oligonucleotides. They are similar to DNA, but they are way shorter. So basically they are the part of the DNA. That's why we call them oligonucleotides. So designing the primer is an important topic, because for a successful PCR reaction you need correct primers. So if you know how to design the primer, what is the mechanism of primer binding, you can design correct primers. So suppose this is the DNA molecule, and as you always know that the DNA molecule is made up of 2 strands. They are coiled together. For convenience, I'm just showing them in the form of a ladder. Now once you heat that particular strand, this is the first step of PCR reaction, which is denaturation. In presence of heat, the hydrogen bond between these 2 strands is going to broken up, and then these strands, they are available for the primer binding. Now what will happen, that you will have 2 different types of primers. Primer 1 and primer 2. Primer 1 will bind to one side of the DNA strand, and primer 2 will bind to the other side of the DNA strand. And here, as you can see, the green colored regions, they are the primers, and that's why I'm going to write primer 1 in this side, primer 2 in that side. And next, what is going to happen is using the DNA polymerase enzyme, extension is going to happen, and then you will get 2 new strands which are complementary to the old ones. So this is the one cycle of your PCR reaction. Now imagine that repeated cycle of denaturation, extension, annealing, extension is going to happen, and then you'll have multiple copies of DNA molecule. And once you have multiple copies of DNA molecule, you can do a lot of different things with these DNA molecules, and that is basically the application of different types of applications of PCR reaction. Now you will synthesize millions and billions. You can basically synthesize millions and billions copies of the DNA molecule, specific region of the DNA molecule. That is very, very important. So as you can see here, in the final stage, you will have lot of DNA molecules, and then you will store your PCR reaction, and further it can be analyzed using agarose gel electrophoresis. So in this particular region of my video, I've shown you how primers they're going to bind, and I have also shown you that primers, they are important. So what are basically primers? Primers are the short oligonucleotides. They are key to the success or the failure of the PCR reaction. If your primers are good means their properties, all the features that you need inside the primer, is there. So basically PCR is going to happen successfully. If you have wrong primers, then you're going to have trouble. What can go wrong in that case? You will have no PCR reaction, that means the result, the outcome of the PCR is nil, or you will get multiple fragments, that is non-specific binding of the primer can happen. And primer, basically they are designed to the region which is flanking to the target sequence. What I mean by that is, suppose you have a DNA sequence that you want to amplify, you need to know the sequence of the border region. And if you know the sequence of the border region, you need to design short oligonucleotides to those regions, and then only you can amplify the region that you're interested in. So here in circle, that is the region 1, and in second circle is the region 2. Another thing is the primers, they should be complementary to the target sequence. So that is also very, very important. Now suppose if you have a sequence, a typical sequence of DNA molecule starting from the 3' end, and it is extended like GT, and then again you have GT, CTG, AGT, and so on. You want to design the primer and see carefully, this will be the sequence of your primer. It should be the complementary sequence to the target sequence. And then this is the region it's going to bind, and from here the extension is going to happen, right? So another important point is the region that you are interested in, it should not be too big. It should be around 1 kilobase pair. If you want to amplify larger than that, the strategies are available, techniques are available. You can even amplify up to 10 kilo base pair, but a recommendation usually for general PCR reaction is less than 1 kilobase pair. But there are tools available that you can use to amplify larger sequences. Now, length of the primer is another aspect. So how long should be your primer? That is also very, very important, that what should be the length of the primer. And length of the primer should be less than 30 base pair, and somewhere around 15 to 20 base pair. Why is that? Because if you have if you have really, really short oligonucleotides, what is going to happen, that they are going to having non-specific binding. And if you have larger sequences, what will happen in that case is your binding will be enhanced. So that's why you need length of the primer that is optimum. So that's why you design primers specifically, and there are tools available to design the primers. There are software available. You just put in the region that you want to amplify, and you can pick out the primers. You can specifically design manually. You can also design your own primers depending upon all the knowledge that we have discussed here. Basically, you can read the sequence and design your own primers. And we'll also discuss those softwares in our later videos. But as you can see here in this particular video, I tried to show you that there are oligonucleotides primers that are required if you want to amplify a specific sequence. And these primers are basically, they are complementary to the target sequence, specifically to the region flanking to the target sequence. And I have also shown you how you can construct the primer. Just only one example. And the similar way, you'll have another primer for the second sequence. We have also discussed that you cannot really amplify the larger sequence up to 10 kilobase pair. You need different types of polymerases for that, but the polymerases, they are available. Another thing is I would recommend that you use 1 kb as the target size of your gene. You should go less than that if you want to have the successful PCR reaction within 2 to 3 hours with up to 35 cycles. Next, the length of the primer, it should be in between 15 to 20 base pair, which is the standard. So in this video, I hope that I have discussed some of the important points regarding the primers, and in the next video we'll discuss more detailed perspective on various components of the PCR, and I hope those videos are also going to help you to understand PCR better. So if you like the video, then please like and share the video with your friends. If you like the content of this channel, then please do subscribe the channel. Thank you and take care.